Good morning, children.
Good morning, ma'am.
Good morning.
Good morning, ma'am.
Only Vidur has joined. Good morning.
Good morning, ma'am.
Morning, ma'am.
We learned about Human Genome Project yesterday. We just started with Human Genome Project, and the cost of carrying out or the cost of carrying out the sequencing of a single base pair and the whole genome we discussed last class, and it took 13 years. Even though they declared it complete after 13 years. In which year it was started, Gayatri? Which year they launched? In 1990, and it was completed. They declared it completed in 2003, but exactly or the sequencing of chromosome number 1 got completed only in 2006, and it is known as a mega project. Human Genome Project is a mega project, seeing the expense involved in it and the time taken for carrying out the process. It's considered as a mega project, and we started with the different methods used for sequencing the genes, how the genes are identified. So Human Genome Project is meant for sequencing the entire human genome, the entire set of genes present in a human being. And the methods we already discussed, 2 methods are used to identify the genes. Michael, which are the 2 methods used? Michael? Sharon? You were not there yesterday. Yes, Michael. Which are the 2 methods?
Identifying ESTs and sequence and
sequence annotation. Okay, sequence annotation and express ESTS means expressed sequence tags. So for identifying these genes, for carrying out the Human Genome Project, 2 approaches were considered. One was expressed sequence tags and the other one sequence annotation. In expressed sequence tags, they concentrated only on, or it was focused only on sequencing those genes which get expressed. You know that all the genes are not getting expressed. Expressed in the sense only those genes which get transcribed and get translated to form proteins. Only those are expressed genes. So in the first approach or expressed sequence tags, they try to identify or sequence the genes which get expressed as proteins, or which get expressed as RNA, and finally they'll get converted to proteins. Whereas in the next approach, sequence annotation, here the blindly they carried out the sequencing of the entire genome, whether coding or non-coding. So they did not separate coding or non-coding. The entire sequence, they blindly sequenced the entire genome, both coding and non-coding. And after that, after completing the sequencing, they assigned different regions in that sequence with different functions. So we will see that once again. All these we had learned yesterday. The methods involved, or the methods to identify genes, involve 2 approaches. The first approach was identifying ESTs or expressed sequence tags. And the second method, sequence annotation. As I told you, in expressed sequence tags, they concentrated on identifying the genes which get expressed. Getting expressed means, you know that in a gene or in a DNA, exons, introns, all these are there, and all the parts are not getting, the entire segment is not getting transcribed into RNA, and these are not forming proteins. So only those which are getting transcribed into RNA and getting translated into proteins, those are the expressed genes. So only those were sequenced or identified. But whereas in the second approach, this was a blind approach. No separation between coding and non-coding sequences. The entire genome is sequenced and they were assigned functions. So the different regions in the sequence were assigned functions. So this we had learned. So these are the two approaches involved. So the methods for identifying the genes involved two approaches. One was expressed sequence tags, where they concentrated on sequencing only those genes which get expressed as RNA, which get transcribed as RNA. Later, they will get translated into proteins. Whereas in the second approach, you call it a sequence annotation, where irrespective of coding and non-coding, the entire genome is sequenced and they were assigned functions. Different regions were assigned functions. And then genome sequencing, how this was carried out. What are the steps involved? Children, genome sequencing. The sequencing means to decide or determine the order of nitrogen bases. That is known as sequencing. So in sequencing, in what sequence the different nitrogen bases, adenine, thymine, guanine, cytosine, in what order they are arranged in these genes. That is the purpose of carrying out genome sequencing, to find out the order of nitrogen bases. So what are the steps involved? How did they carry out this method? First of all, the entire DNA from a cell, a single cell is enough to carry out the process. The entire DNA from a cell is isolated. It is separated and then it is broken down into different fragments. And the breaking up of fragments is with the help of enzymes known as restriction enzymes. You don't have to learn about it now. In biotechnology chapter, you will learn in detail about restriction endonucleases, exonucleases, etc. So with the help of the enzymes, the DNA, which is isolated from a cell, is broken down and it forms small fragments. Now, these fragments are cloned in a suitable host. We need a host organism. And here they used bacteria as well as yeast. So two hosts were considered, bacteria and yeast, using specialized vectors. And the vectors they used were bacterial artificial chromosome and yeast artificial chromosome. So the host which they used, which they considered, were bacteria and yeast. The vectors which they used were bacterial artificial chromosome, abbreviated as BAC, and yeast artificial chromosome, abbreviated as YAC. And how this is happening, how the cloning takes place. So what is done is, now the DNA is broken up into small fragments. Okay. And then they take this artificial chromosome. The vectors are taken. Okay. These vectors are taken, which will be like plasmid. We have learned about plasmids, no? They will help in transferring the foreign genetic material into the host cell. So here a cut is made and then what is done? The desired DNA is introduced. See, this is the vector, and from vector a small piece will be removed, and then the desired, this DNA fragment will be introduced. This is human being, or this belongs to human being, and this is the vector. Okay, and then it will be introduced into a host organism like bacteria or yeast, and here we'll be introducing it. Okay, then what happens? As this divides, more and more copies of this will be formed. So as they multiply, more and more copies of the human genome or this DNA fragment also will be made. So cloning means making identical copies, forming identical copies using specialized vectors. Did you understand, children? I'll repeat again. First of all, to begin the process, the DNA, the entire DNA is separated, is isolated from the cell. And then with the help of the enzymes, these are broken down into small fragments. They are broken down into smaller fragments. Then what they did, we need to make multiple copies of each fragment to make the sequencing process easier. So for that, they selected host organism like bacteria or yeast. A host organism is selected, and then they did this with the help of vectors. And vectors, usually about plasmids, you have learned extra chromosomal circular DNA, which will help in transferring foreign DNA into a host cell. So let this be the vector. Okay, that is also DNA, genetic material. And then what is done? We will insert it into, this can be inserted into this vector. From here, one portion will be removed. A portion of its, this vector will be removed, and we will be inserting human, the fragment of human DNA. Then what is done? This will be introduced. This will be introduced into a host organism. And as the host organism multiplies, reproduces, we get more and more copies of this particular fragment. So when we get more fragments, it will make the sequencing process easier. If you are having just one fragment, and if somehow it is getting damaged, we will have to start the process from the beginning. So here we are making multiple copies. We are cloning. We are making identical copies of the same, or we are carrying out amplification. Means many copies are being made. Okay. So these fragments are cloned in a suitable host using specialized vectors. So they amplify in the host. These cloned fragments, as the host organism increase in number, these fragments also will amplify, or multiple copies will be formed. So when it is amplified, it makes the sequencing process easier. So same fragment, we are having so many copies, it will be easy to carry out the sequencing process. Then the common vectors used: bacterial artificial chromosome, yeast artificial chromosome. The host which we use for carrying out are bacteria and yeast. Now amplification has taken place. Each DNA fragment had been made, or several copies had been made. Now what is next? Automated DNA sequencers. Automated DNA sequencers are used to sequence these small fragments. So from why we are breaking it into smaller fragments, you know that the DNA is around 2.2 meters. It is not that easy to sequence the entire DNA at a stretch. So in order to facilitate easy sequencing, it's broken down into a split into several fragments, and then these fragments are sequenced. It is not easy to do it humanly, so machines are used. Automated DNA sequencers are used. These automated DNA sequencers, it work on the principle of a method developed by Frederick Sanger, and that is why we call the process as Sanger sequencing. Frederick Sanger. It works on the principle of a method proposed by Frederick Sanger, and you call this method as Sanger sequencing. Then once the sequences are obtained, they are arranged based on the overlapping regions. Yesterday we learned what are overlapping regions. Okay. And overlapping regions within them. Then the next process is alignment. Once again, using automated DNA sequencers, we determine the order of nitrogen bases in these smaller fragments, and then the next process is aligning them. on the basis of overlapping regions, and this is done with the help of computer programs or with the help of specialized computer-based programs. So it is not possible to do it or not it down in a notebook and all. About bioinformatics we learned in the last class. So with the help of computer-based programs, all these sequences are aligned, arranged. How are they arranged? Based on the overlapping regions. And later, these sequences are annotated. To annotate means to determine their location and also find out its function. So after the sequences are arranged with the help of computer programs, then each sequence or the different regions of the sequences are annotated. Their location is identified and their function is determined. What function do they perform? And then it is assigned to each chromosome. You know that there are different chromosomes. So this particular gene or segment or DNA sequence, it is part of which chromosome? We have 23 pairs of chromosomes. And into which particular chromosome this sequence belongs to. So all that will be determined after that. It is not as easy as we say, but these are the basic steps involved. So quickly I will say once again. First of all, the entire DNA from a cell is isolated. And then what is next? It will be broken down into small pieces, small fragments, because it is not easy to study or to sequence the entire DNA, which is 2.2 meters in length. So first it is isolated, then it is broken down. Next step is using specialized vectors. Vectors are carriers, or these will help in transferring a foreign genetic material into a host organism. So using vectors, we transfer this into a host cell, and inside the host cell, cloning takes place, means identical copies will be formed. So when several copies of this are formed, as and when this host organism multiplies, more and more copies of these genetic material will be formed. This will lead to amplification of the DNA fragments. Means amplification means several copies, making several copies. It's getting amplified. This amplification is necessary because if it is just a small fragment, scientists will find it difficult to carry out studies because just one they have. When several copies are made, it is easy. It will facilitate the sequencing process. And the common vectors used are BAC and YAC, and the host organisms used were bacteria and yeast. Then once these sequences are amplified, next step is to sequence them, to find out the order of nitrogen bases present in them. That is done with the help of machines known as automated genome or DNA sequences. Automated DNA sequences. We call this method as Sanger sequencing also, because this works on the principle of a method suggested by Frederick Sanger. And then once the sequences are obtained, it will be arranged on the basis of overlapping regions. It will be arranged on the basis of overlapping regions, and the arrangement or alignment, that is also done with the help of computer programs. Once that is completed, then annotation is done. Annotation means to identify the location where this particular sequence is present, in which chromosome, what function they perform. All that is decided or determined. And for those who missed yesterday's class, I'll just tell you what is meant by overlapping regions. See, if this is a segment, here you can see A, T, C, G, G, C, T, A, like this. Okay, let this be a sequence of nitrogen bases. And you know that 3 of them will form a codon. So this is This forms one codon. Usually it is like this, no? This forms another codon, this another codon, and thereby it continues. But just see if it is like this. ATC forming one codon, CGG forming another codon. So that means here there is an overlapping. ATC is forming the first codon. Next codon is CGG. That means C is common for these two codons. So there is an overlapping taking place. So there are, or you can say less than 14%, 13.36% of the DNA has got overlapping regions. In human genome, there is almost 13.36% of overlapping region. So there this happens. It can be two also, not just one. Say for example, if this is CTA and the next sequence can be ATC or it can be TAT also. So wherever that overlapping is taking place, based on that, the sequence is arranged. Once again, I will show it. A, T, C, G, G, C, any order. Okay. And normally what you have learned is this is one triplet codon, another one, another one, another one. This is that what we have learned. But wherever overlapping is taking place or wherever overlapping sequences are there, it could be like this. ATC forming one codon. The next codon can be CGG. And maybe the other one can be GGC. So overlapping is taking place. And certain nitrogen bases are common for two codons. So when this happens, you call it as overlapping. So once the sequencing is done, they are arranged on the basis of overlapping regions, and their alignment is also done with the help of sequencing. I told you sequencing means determining the order of nitrogen bases present. So sequencing as well as their arrangement, their alignment is also done with the help of computer programs. Once it is done, their location is determined or identified and function is decided, and it is assigned to each chromosome. This particular sequence belongs to which chromosome? And the sequencing of chromosome number 1 was completed in May 2006. The project was launched in 1990 and they declared it complete in 2003, but actually some work was pending and it was completed in 2006. Actually the sequencing of chromosome number 1 was pending, which they could complete by 2006, May 2006. Now, assigning genetic and physical maps on the genome. And that was also another task which the scientists had to carry out, assigning genetic and this is given very briefly in your textbook. Another Herculean task which they had to do was to assign genetic and physical maps, prepare physical and genetic maps on the basis of human genome. And this they prepared with the help of the information, they had on polymorphism of restriction endonuclease recognition signs, and using the information available on some repetitive DNA sequences known as microsatellites. Only this is given in your textbook regarding the mapping process. I'll repeat once again. As part of the human genome, they had to prepare genetic and physical maps. And physical map is actually meant to find out. So the difference between genetic and physical map is that in physical map, the order and the physical distance between the DNA base pairs are found out, or that is plotted. Once again, in physical map, the order as well as the physical distance between the nitrogen base pairs are determined. Whereas in genetic maps, they give us the information regarding in which chromosome a particular gene is present, and exactly in the chromosome where the genes are present. All those information we get from genetic maps. You don't have to learn the details of genetic and physical map. Only the terms are mentioned. Once again, I will say physical map will help in finding out the order and the physical distance between the nitrogen base pairs on DNA. Whereas genetic maps give us information regarding where specific genes are present or which chromosome is having that particular gene and where they are located in that chromosome. All the information we'll get with from this genetic map. So genetic and physical maps, and scientists prepared it using the information on polymorphism. Poly means several, different. Morphism, more forms, a genetic variability, that is known as polymorphism. So in among different organisms, the character is expressed differently, the features expressed differently. Here it is genetic variability based on the polymorphism of restriction endonuclease recognition sites. Restriction endonuclease, earlier I told you, DNA is isolated, then it is broken up into several fragments, and enzymes are used for the process. So this is the enzyme. Restriction endonuclease is the enzyme used for cutting the DNA into fragments. And these enzymes, they cannot cut the DNA anywhere they like. There are special sites. They will recognize those sites. There only they will cut the DNA fragment. DNA cannot be cut into fragments at any place or at any location. These enzymes will recognize specific sites. There only they will cut the DNA fragment. So those sites are known as restriction endonuclease recognition sites. And these sites may not be identical in different individuals. It can be different for different human beings. So the polymorphism, based on the polymorphism of restriction endonuclease recognition sites. Once again, restriction endonuclease is the enzyme which will help in cutting the DNA into fragments or pieces. And they cut the DNA at specific sites, not anywhere. There are specific sites which these enzymes will recognize and cut it into fragments. So that recognition sites are different in different people. So the polymorphism of restriction endonuclease recognition sites, and based on microsatellites. Microsatellites are DNA sequences which gets repeated. They keep on repeating. So in our DNA, certain sequences, they keep on repeating. Those sequences are known as microsatellites. So here A, T, C, G, G, C, A. Maybe this is the particular sequence. This can get repeated again. A, T, C, G, G, C, A. Okay. The number of times it gets repeated can be different. The number of times it is getting repeated can be different. So these some repetitive sequences are present in the DNA. In everyone's, in all human beings, genetic material or genome, some of the sequences will get repeated. And these repetitive sequences are known as microsatellites. So based on genetic and physical maps are prepared by scientists based on the information regarding polymorphism of restriction endonuclease recognition sites and microsatellites. What are microsatellites? These are some repetitive DNA sequences. That sequences gets repeated at certain parts of a DNA. Now, what are the salient features, important features of human genome? Human genome contains 3 into 10 raised to 9 nucleotide bases, 3,164.7 million. In number, if you say 3,000 164.7 million nucleotide bases are present. So by carrying out human genome project, they found out these many bases are there. An average gene, if you take, gene means it's a segment, a piece of DNA. So the number of nitrogen, the length of the gene can be different. Depending upon the gene, the length can be different. All genes are not of the same size. So on an average, it will have 3000 bases. On an average, 3000 bases are present in each gene. However, the size of the gene differs. As I told you, we have how many genes? That also is mentioned here. Several genes are present, and all these genes, 30,000, almost 30,000 genes are present, and all the genes are not of the same size. And each one is having approximately 3000 bases. The largest gene, these are the information which they collected, which they got from carrying out the project. The largest one, the largest gene is dystrophin. Dystrophin is having 2.4 million bases, whereas on an average, I said 3000 bases are present in a gene. Depending upon the length of the gene, the number of bases present also can differ. So the largest gene is known as dystrophin, 2.4 million bases. Total number of genes in human genome is 30,000, and 99.9% of the nucleotide bases in human beings are same. Only 0.1, that makes all the difference. 0.1% makes all the difference. 99.9% of the nucleotide bases, you can say 99.9% of the DNA is same. DNA is same in all individuals because our basic functions are same. The different metabolic processes taking place in our body are same. So basic functions are same. At the same time, we are unique. We have some features that distinguishes us from other organisms, which makes me and you different. That is just 0.1%. Remaining 99.9% is same. Over 50% of the discovered genes have unknown functions. So we said 30,000 genes are there, but scientists haven't succeeded in finding out the functions of 50% of them. Less than 2%, just less than 2% is coding for proteins. Okay, only less than 2% of the genes are getting expressed to form proteins. Large portion of genome consists of repeating sequences. The sequences are getting repeated. A large portion, in a large part of the human genome, the sequences are getting repeated. I'll show it once again. Human genome consists of 3 to 10 raised to 9 nucleotide bases. An average gene consists of 3000 bases, but this can vary depending upon the gene. But on an average, 3000 bases are there. The largest gene is dystrophin. It has got 2.4 million bases. The total number of genes present is 30,000, and 99% of the nucleotide bases is same in all individuals, and only 0.1% makes the difference. Over 50% of the discovered genes have unknown function. So far, the scientists have been succeeded in finding out the functions performed by those genes, and less than 2% of the genome codes for proteins. And large portion of the human genome consists of repetitive sequences. Okay. Repetitive sequences have no coding function. They are non-coding. Wherever the sequences are getting repeated, in some parts, many parts, they said that large part of the genome is having repetitive sequences. So they are not having any coding function. Already we learned less than 2% of the genes are coding for proteins. They don't have any coding function. Sometimes these sequences can get repeated over 100 to 1000 times. That repetition can come up to 1,000 times. And these repetitive sequences can have a role in evolution, chromosome structure, and even in dynamics. Chromosome having the maximum number of genes is chromosome number 1. Maybe that was the reason it took extra 3 years for completing its sequencing. 2,968 genes are present on chromosome number 1. And the largest gene is dystrophin because it's having 2.4 million bases, and the chromosome with the maximum number of genes is chromosome number 1, 2,968. Chromosome with the least number of genes, chromosome number Y, just 231. All the other genes, all the other chromosomes are having more than 231 genes. SNP or single nucleotide polymorphism, it is pronounced as SNPs. SNPs or single nucleotide polymorphism occurs at about 1.4 million locations in human DNA. Single nucleotide polymorphism means in a particular sequence, A, T, C, G, G, A. Okay. In person 1, it is like this, and person 2, it can be A, C, G, G, A. And maybe in the third person, it can be A, it is G. That means single nucleotide polymorphism. A single nucleotide, there are changes. A single nucleotide polymorphism means it is affecting just one nucleotide base. So in person 1, there it is thymine, whereas in the second person, it is C, polymorphism, genetic variability. Accordingly, the functions performed also can change. And in the third person, it is G. One person it is T, the other person C, and another person G. So single nucleotide polymorphism, it affects a single nucleotide base, and it is found at about 1.4 million locations in human DNA. This information has revolutionized the process of finding chromosomal locations for disease-associated sequences and tracing human history. So the information regarding single nucleotide polymorphism is very important in carrying out researches related to diseases and also in tracing human history. Single polymorphism means a genetic variability. Single nucleotide polymorphism is the one which is affecting only a single nucleotide. Such single nucleotide polymorphisms can be seen at 1.4 million locations in human DNA. So with that, we have completed Human Genome Project, and next is about DNA fingerprinting. You might have heard about DNA test, DNA fingerprinting, paternity test using DNA fingerprinting. So just like no two individuals can have the same fingerprint, similarly, the DNA of two persons cannot be exactly identical. As I told you, 99.9 will be same, but the 0.1%, that will be different for different person. And that is making us, all of us, different from each other. DNA fingerprinting is a quick method to compare the DNA sequences of any two individuals. So we can compare the DNA sequences of two individuals with the help of DNA fingerprinting. 99.9% of the base sequences in all human beings are identical, no differences. But the difference is only in 0.1%, which is making each one unique. And it is very difficult, expensive, and time-consuming process to sequence and compare. So you know that we are having 3 into 10 raised to 9 million. Sorry, 3 into 10 raised to 9 bases. So what is done? If you need to compare two individuals, the DNA of two individuals, if you have to compare the entire genome, then it will be very difficult, expensive, and it takes time. So it is better to compare those repetitive DNA sequences which makes the difference. Once again, 99.9% of the base sequences in all human beings are same, only 0.1% is different. So if you want to compare the DNA of two persons, and if you are going to compare the entire genome, it will be very time consuming, expensive and tedious task. So what can be done? Certain specific regions known as repetitive DNA sequences are compared, are used for comparative study rather than concentrating on the entire genome, because 99.9% is same. So where the difference? Only 0.1%, and the difference lies in these repetitive sequences. So how many times it is getting repeated, that will be different for different individuals. So there are certain repetitive sequences, and the number of times these sequences are getting repeated can be different for different persons. So here you can see examples. GAAC. Again, GAAC. 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 It is repeated 4 times. GAAC, 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 5 times. GAAC, GAAC, like this, it is repeated 8 times. So the number of times it is getting repeated is different. So by comparing those repetitive sequences, we can compare the DNA of two different persons. We don't have to study or compare the entire genome or entire DNA. Okay, we'll learn more about it in the next period. We have one more period, so we'll learn. So I couldn't ask you any questions because we have less time. So from next day onwards, we are going to do only revision. I don't know whether you have understood. Some of you missed yesterday's class. I don't know what is the reason. I thought you might have you come to the school. That's why you missed it. I had repeated the basic points which I discussed, the methods, the 2 approaches involved. I had discussed. The topic is difficult. So you have to be attentive, and next period also, it's an extra period. So please don't miss the class. Once you miss it, you'll find it difficult. Just by reading the textbook, you will not be able to follow it. So please attend the class. By next period, we'll complete the topic. Okay, children. Please don't forget to join next period. Thank you.